News release, Tuesday, February 21, 2023. Newly discovered gene helps some yeast endure toxins and can help scientists understand toxin resistance. National Institutes of Health researchers have identified a gene that makes yeast resistant to a lethal toxin, according to a new study published in the Proceedings of the National Academy of Sciences. To study the evolution of toxin resistance, researchers at the National Human Genome Research Institute NHGRI, part of National Institute of Health, used yeast of the kind commonly used for home baking as a model organism. While researchers have long known about yeast's remarkable ability to evade the effects of lethal toxins, the reason was a mystery until now. The intricacies of genomics that mediate these within-species battles are beautifully revealed by a study like this, said Charles Rotimi, Ph.D., scientific director of the Intramural Research Program at NHGRI. While this is a yeast story, the mechanisms will surely influence studies on toxins and their effects on humans. Throughout human history, people have combated various toxins made by other organisms, like spiders, plants, snakes, and even the cholera or anthrax bacteria. Understanding toxin resistance in yeast could lead to new avenues for protection against toxins in humans. We're interested in understanding how genomic variation leads to differences between individuals, so in this study, we're looking at the most basic biological mechanisms underlying resistance to toxins in simple organisms such as yeast, said Merusedho Ph.D., an investigator in the genetic disease research branch at NHGRI and senior author of the study. An important way that organisms vary is in how much they are affected by toxins. Typically, toxins are used in conflicts between different species. However, these killer yeast are interesting and safe to study because the toxins only affect other yeast and do not harm humans, said Ilya Andreev, a former NHGRI post-baccalaureate trainee who led this study. There are very few examples of these within-species conflicts in nature, and our work has just scratched the surface of understanding the evolutionary dynamics of such conflicts, Andreev added. In this current study, NHGRI researchers analyzed yeast infected with a virus that causes the yeast to secrete a lethal toxin called K28. The virus does not negatively affect the infected yeast. Instead, infected yeast are also resistant to the toxin's effects. These infected yeast secrete the K28 toxin to wipe out non-infected yeast growing nearby. This provides the infected yeast an evolutionary advantage in the competition for resources. However, some non-infected yeast grow despite the presence of the toxin. To find out how these non-infected yeast resist the toxin, the researchers exposed different non-infected yeast to the K28 toxin. Those unaffected by the toxin were classified as highly resistant and the affected ones as sensitive. Then, the researchers compared the genomes of resistant versus sensitive yeast to identify which genes caused some yeast to be resistant. Through this investigation, the researchers determined that the KT-DUN gene provides resistance to the K28 toxin. This gene has never been studied before, said Sethu. Identifying this gene is the first step to fully understanding what is happening on the molecular level. The researchers then attached a glowing protein to the KT-DUN protein to track its position in the yeast cells. They found that the KT-DUN protein resides on the surface of cellular compartments called vacuoles. Vacuoles serve many purposes in the cell, including isolating and breaking down harmful substances like toxins. To inflict its toxic effects, the K28 toxin must move freely around the cell. The researchers hypothesized that the KT-DUN protein may be involved in capturing the toxin in the vacuole. A region of the KT-DUN protein pokes into the center of the vacuole, where it could interact with trapped toxins. By analyzing the protein sequence, the researchers found that this region of the KT-DUN protein is under strong evolutionary pressures. These strong evolutionary pressures point to this region's importance for the KT-DUN protein's function and highlight the competition between the toxin and the yeast. However, more research is needed to understand how this region of the KT-DUN protein keeps K28 in check and how K28 may evolve in response. NIH is one of the 27 institutes and centers at National Institute of Health. The NIH Division of Extramural Research supports grants for research and training and career development at sites nationwide. Additional information about NIH can be found at www.genome.gov. About the National Institutes of Health. National Institute of Health, the nation's medical research agency, includes 27 institutes and centers and is a component of the U.S. Department of Health and Human Services. National Institute of Health is the primary federal agency conducting and supporting basic, clinical, and translational medical research and is investigating the causes, treatments, and cures for both common and rare diseases. For more information about National Institute of Health and its programs, visit www.nagov. National Institute of Health Turning Discovery into Health Registered.